everyone. And first of all, I'd like to thank Rami and Yoni for the invitation. And it's a privilege to be here, and I'm truly happy. And yesterday has been a very exciting day, although it was mostly clinical. But it's always nice to join the clinical and the preclinical aspects. So this was actually great. And again, thank you. So we're going to switch a little bit gears because I'm going to show you some data of a project that we have in the lab that actually is the exposure to predictive risk factors for vulnerability to initiations and experimentation with drug use, in particular with tobacco and nicotine, and with tobacco and alcohol and cannabis. That's what's shown in humans. But we use rodent models, and this is a project that we have in the lab where we actually look at, where we try to address my main research question, which is an effort in understanding the mechanisms underlying vulnerability and resilience to neuropsychiatric disorders just in general. So in my lab, we focus on a particular window of development because that is pre-adolescence, because data shows that and I do this, okay. Data shows that a large proportion of people affected with mental disorders, they are diagnosed very early on in life, and as a matter of fact, by the age of 14 years. And this is really, for me, it's it's really concerning and somehow scary. And while one would think that disorders diagnosed so early in life are disorders of neurodevelopment, which show remarkable sex dichotomy, actually data shows that disorders other than neurodevelopmental ones are diagnosed very early in life. And these include mood disorders, substance use disorders, eating disorders, OCD, and anxiety. And since I'm interested in the left tail of these curves, of course, then this is highly relevant because these disorders are diagnosed in vulnerable individuals before the actual medians for the single curves. So what we know about on the origin of mental disorders, according to the most influential theories, is that this depends upon the interaction between environmental risk factor, the exposure to environmental risk factors, and the biological makeup of the individuals. And developmental vulnerability is often restricted to early life moments when this interaction does occur, and this would bias some developmental trajectories to lead the transition from the endophenotype to the onset of mental disorders. And what we know is that these endophenotypes are often associated with some changes, subtle changes in behaviors, and these are often associated with changes, alterations in the function of dopamine neurons, which are then sensitized, and they would respond in an aberrant manner to subsequent insults later on in life, and this leads to misattribution of salience to otherwise irrelevant stimuli, and this would bias the cognitive schema that an individual uses, further biasing the altering the cognitive process of the individual. So what we know is that among the several predictive risk factors that can alter the individual's life early on in life is maternal lifestyle. And this has been shown to change the endocannabinoid system not only of the mother but also on the fetus. This is particularly relevant because it has been shown that endocannabinoid system plays a pivotal role throughout development and also during adulthood. But if you look at this figure where the levels of the different molecular determinants of this endocannabinoid system are depicted, you can see that this change importantly throughout development and in particular during gestation. So if you interfere with even one of these molecular determinants, and you can actually alter the endocannabinoid system, and therefore all the processes, or even one of the processes that this system is involved, and therefore this can impact on proper brain maturation, as such as said before, proper is the norm, right? That is important. There is, I know that everyone is pretty much familiar with the endocannabinoid system, but I'm just going to say that for those that are not familiar, that endocannabinoids, according to the canonical models, they are produced and released on demand by the neurons at the postsynaptic level, and this demand is basically an increase in postsynaptic calcium that can be driven by either activation of a receptor or depolarization of the neurons. And this will lead to the release of endocannabinoids, which would move retrogradely across the synaptic cleft and bind in the brain mainly to type 1 cannabinoid receptors, which through a variety of mechanisms lead to inhibition of neurotransmitter release. And this way of functioning is actually the way that endocannabinoids can shape the strength of neuronal synapses in both short and long-term manner in the brain, and there is so far no exception to this rule. So a way that we can enhance endocannabinoid signaling is through direct activation of CB1 receptors, for example, by using cannabis through the main ingredient of cannabis, that is THC, or indirectly by enhancing endocannabinoids, and this would occur through inhibition of either degrading enzymes or inhibition of the putative transporter. But for the sake of this talk, I'm going to focus on the actually direct agonism and the THC and the exposure to THC during the prenatal life, because there is a concern among neuroscientists, but also among physicians, about the use of maternal cannabis during gestation. And this is because it's been on the sharp rise. The prevalence rates have been on the sharp rise for the past 1 decade or maybe 15 years, and probably due to the expanding legalization and liberalization policies. And this led the general population, and in particular pregnant women, that cannabis is a safe compound, and because it's safe, it is safe to use it even during such a sensitive period that is pregnancies. And actually among the general population and among pregnant women, the perceptions and the knowledge of the risks that are associated with cannabis use for the fetus are minimal. If none at all. And this is despite the wealth of epidemiological studies showing that there are long-term detrimental impact on both cognitive and behavioral domains for the offspring, and that is the reason why there is a consensus on how important it is to generate more data, especially in humans with these longitudinal studies. But with these preclinical studies are pivotal because, and they are instrumental to disentangle the different components of these effects. And in fact, animal models have dissecting the effects of a single ingredient of cannabis, let's say CBD versus THC, then you can establish a dose dependency, you can establish the time dependency, and more importantly, you can establish if there is one developmental time window that is more sensitive to the other to the exposure of cannabis. So about 10 years ago now, we have developed a rat model of prenatal cannabinoid exposure that is the one that when we passively administer THC for 2 weeks during gestation, mimicking the exposure during the first and the second trimesters, because usually women then that use cannabis said they would decline the use during the third trimester to start back when they're breastfeeding. And we use a dose of THC that actually does not affect the dam behavior. These are Sprague-Dawley rats, and is not teratogenic, meaning that it does not affect the litter size, and it does not affect the male to female ratio. We also found that this does not affect maternal and non-maternal behavior, which is very pivotal for proper maturation, emotional maturation of the offspring. And then we perform our analysis during the pre-adolescent period for the reasons that I mentioned at the very beginning of my talk, and we perform a multi-scale analysis. And so what we have found is that PCE, which is prenatal cannabis exposure from now on, going to turn as PCE, induces a male-specific hyperdopaminergic phenotype that is associated dopamine neurons of the ventral tegmental area, actually those located in the lateral portion of the posterior VTA, which project to the lateral shell, and so they have a particular encoding signal. And this is associated with an increase in the excitability of pyramidal neurons of the deep layers of the medial prefrontal cortex. And we also found that there is PCE induces a male-specific imbalance between excitation and inhibition in both brain regions, so the VTA and the medial prefrontal cortex. And this is particularly relevant because an imbalance between excitation and inhibition in the prefrontal cortex is associated with risk assessment. And what we found is actually that there is a male-specific reduction in risk assessment in these animals. This is also associated with deficits in social playing behavior, which strongly depend upon the function of the prefrontal cortex. And we also found, but this is all published, that these animals they exhibit poor coping strategies when exposed to an acute unescapable stressor, and this would actually sensitize them to deteriorate their sensory motor gating function, an effect that again is man-specific. Now, sensory motor gating functions are the ability that individuals across species have to filter out irrelevant stimuli. So they focus on what is salient, and they discard those stimuli that are not salient, that are irrelevant, irrespective of the valence. And you understand that this is a really important function for species survival, right? So what we found is that this deterioration, this sensitivity, actually relies on the hyperdopaminergic profile that we have observed in our VTA. And in fact, when we chemogenetically normalize the activity of these neurons before the exposure to an acute unescapable stressor, we're able to fully restore proper sensory motor gating functions. Conversely, when we drive hyperdopaminergia in the females, which are usually resistant, then we are able to, before the exposure to stress, then we are able to deteriorate this sensory motor gating function, thus suggesting that it is actually the hyperdopaminergic phenotype that is driving this sensitivity, this vulnerability to stress. Now, given that we know that acute stress potentiates excitatory synapses on VTA dopamine neurons, an effect that be counterbalanced by 2AG, by the endocannabinoid twerk glycerol, we considered possibility that this signaling could be affected by prenatal cannabis exposure. And an easy way as electrophysiologists to assess the proper function of 2AG phasic signal in the VTA, on VTA dopamine neurons, is by assessing a type of plasticity, a short-term plasticity that these dopamine neurons express upon duration of depolarization of at least 5 seconds. This is termed depolarization-induced suppression of excitation, in other words, DAC, and we have shown more than 20 years ago that this requires the synthesis and the release of 2AG and the activation of presynaptic cannabinoid type 1 receptors. So we decided to explore whether this plasticity could have been affected, and indeed we found that PCE affected both sexes. In fact, it is abolished, DAC is abolished in females, it is enhanced in the males. This is associated with changes in the presynaptic, in the pair pulse facilitation, consistent with the changes in the plasticity that suggested that these are changes in presynaptic locus. And so then we, of course, wanted to check whether this was mediated, again, by CB1 receptors. And when we pharmacologically blocked these receptors, we found that CB1 was mediating, DAC was mediated by CB1 receptors in all groups, but in PC male dopamine neurons. So at this point, we said, okay, so maybe these receptors are differently working, or maybe they are different in their expression. And so an easy way, again, because we are electrophysiologists, to assess this is to build those response curves for the CB1 receptor agonists. And that's basically what we did. We built those response curves for two structurally different CB1 receptor, oops, I forgot to say, okay, that this type of plasticity in males is it displays a different time curves that is very different from the control animals and what we have published in rats and also C57 black mice. And it actually features a type of long-term plasticity, okay, long-term depression. So we wondered again whether CB1 receptors were involved in this. It seems that it appears that the induction of this plasticity is not affected, as I showed you earlier in the DSC. And so, again, we built those response curves, and we found that in PC male dopamine neurons, these agonists, CB1 receptor agonists, fail to reduce the probability of glutamatergic release. Glutamate release, and this effect does not depend on the number of CB1 receptors, and this has been done in collaboration with Ishvan Katona, but it depends on the fact that these receptors are located farther away from their effectors, which is voltage-gated calcium channels, which is taken as a proxy with the active zone protein bassoon. And this means that when you depolarize the dopamine cells, or when you apply a CB1 receptor agonist, would prevail excitation of dopamine neurons instead of the inhibition that we usually observe. So at this point, just to remind you, we are depolarizing the dopamine neurons to induce this type of plasticity. And given that 2AG cannot work, clearly, because these receptors are located farther away at the excitatory synapses, the possibility is that an alternative retrograde neurotransmitter is released upon this depolarization. And actually, Julie Cower has published a number of interesting papers showing that tetanization of excitatory synapses on dopamine neurons can induce a release of nitric oxide that would induce heterosynaptic form of long-term potentiation. And so we decided to test this possibility, of course, as electrophysiologists and pharmacologists, and that's the easiest approach that we took. And then we decided to block nitric oxide synthase in the slice with a name, and then directly in the dopamine neurons through the patch pipette. And what we found is that this pharmacological manipulation was able to fully prevent DAC in the PCE male dopamine neurons without affecting, more importantly, the DAC induction and the expression in the control dopamine neurons. And then given that NO actually activates the soluble guanylate cyclase, and Julie Cower has shown that this type of plasticity is mediated by an NO cyclic GMP signaling, we decided to test the possibility that, again, these molecular components was actually involved in the signal and in the plasticity that we were observing, and that's basically what we found. Again, a similar effect. So basically, nitric oxide cyclic GMP signaling is involved in this long-term depression of excitatory synapses. We next decided to test how much NO is released by dopamine neurons, because there are some conflicting evidence of how much this retrograde messenger can be released by dopamine neurons. So we took advantage of a calcium permeable fluorescent probe for NO, and we found that upon 5 seconds depolarization, there is a 5-fold increase in NO in the VTA slices of PC male dopamine neurons, and this is associated with an upregulation of neuronal NO synthase in TH-positive neurons, and again with an increase, a massive increase in cytosolic calcium on dopamine neurons, again exposed in utero to THC. Now, at this point, the scenario appears that upon 5-second depolarization, dopamine neurons, which already have an increase in cytosolic calcium, they would release an O that would probably activate, induce this type of LTPI, and probably this massive release of GABA could spill over into neighboring excitatory synapses to activate GABA B receptors and probably mediate this type of LTD at excitatory synapses. So we test this possibility. I'm almost done, Rami. We test this possibility, and indeed we found that we could observe LTPI only in male dopamine neurons of PCE animals. This is associated with a per-pulse depression that's suggestive of an increase in the probability of GABA release, and this effect requires the synthesis of NO by dopamine neurons because it could be blocked pharmacologically. We also found that this LTD, which I show you, relies on NO cyclic GMP signaling, is indeed mediated by GABA B receptors, and given that this is a type of, rearrangement of homeostatic changes, we thought that this could confer, this switch in neurotransmitter, retrograde neurotransmitter release could confer some circuit stability. In other words, could confer long-term depression at synapses that, as we have published, they undergo metaplastic changes by inducing, which are that you can induce LTP even for low frequency of stimulation, and then we consider the possibility that this LTP could be occurring because some GABAergic synapses are less functional. And because Julie Cower has shown that this NO cyclic GMP signaling does occur in specific circuits, we, particularly in the nucleus accumbens projection, direct projection to the dopamine neurons, we decided to test the possibility that actually PCE induces some circuit-specific rearrangement of the inhibitory architecture in the VTA. And so for the sake of this talk, I'm just going to show you two data where we basically, by using viral strategies, we optogenetically interrogate two circuits, the one from the nucleus accumbens, for the reason that I just told you, and the other one from the rostromedial tegmental nucleus, because we have shown 15 years ago that this powerful controls the spontaneous activity of dopamine neurons. So this is going to be quick. So we use an optogenetic approach and found that the control operated by the nucleus accumbens inputs on BTA spontaneous activity is markedly reduced in the PCE male dopamine neurons. This is associated with an increase in the failure rate. Actually, usually these synapses do not show any failure rate, so they're very effective. But there is a failure rate, and this is associated with a reduction, again, a sex-specific reduction in the connectivity between these inputs and dopamine neurons. And when we look at, and this is an effect that is sex-specific, again. So when we look at the RMTG nucleus, we found, again, a reduction in the control that RMTG nucleus inputs have on the dopamine firing rate. There is an increase, again, a massive increase in the failure rate, which is associated with larger facilitations at these synapses, and that's suggestive of a reduction in the probability of GABA release. And as you can see here, when you optogenetically activated these inputs, then you have a complete silencing compared to the controls in this input. And so that is the reason why we went and looked at the spontaneous activity of the RMTG GABA neurons and found that when there is no changes in connectivity, there is a reduction in the spontaneous activity of dopamine neurons. So, and again, this is an effect that is male-specific. So just to conclude, and I hope that I'm on time now, I hope that I convince you that prenatal cannabis exposure in terms of prenatal THC has remarkable sex-specific effects. It can induce changes in the way that endocannabinoid signal at different synapses. In particular, it does disrupt the way it works at excitatory synapses, and this is associated with changes in the architecture and circuit-specific changes in the architecture of the inhibitory inputs on VTA dopamine neurons, and this is associated with endophenotypes that are at risk for psychopathological traits. And with this, I want to thank all the people that in my lab work on this project, some other collaborators in my university and across the globe, of course, funding, and you all for your attention, hoping that I'm on time.
Yes,